So when we finished last time, we had started talking about concentrations of molecules in solution, and as it relates to what you can and what you shouldn't do for and with a patient. And so as we made our way through looking at calculating concentrations of solution, understanding the differences between molarity and molality, which is very little difference. Both represents how many molecules of a solute are in a solution. One is in a total volume of a liter, no more than, and that's molarity. The molality is a certain number of molecules in exactly 1 liter added to it, which means your volume is going to be more. So you see there's not much difference. Now, Megan told me in SI that a lot of people are really sweating, oh, if I got to calculate molarity. No. I'm explaining the definitions of that to help you understand as health practitioners and nurses that you have to know the osmolality of a patient's blood for the times that you give them IVs. You're giving them solutions that contain solutes. In this case, our dextrose, that's a solute of carbohydrate. In this case, we have a solution of saline. Your solute is sodium chloride. And it's imperative that you give the patient the exact same concentration of solutes in a solution. Remember, we used that freezing point depression of the plasma to determine that the osmolality of human plasma is what? What number? Do you remember that? It's the same as the solutions you're going to put in your patient. 300 milliosmoles. So going through all that molarity, molality, knowing the definitions for those will be important. But the whole point of doing that was to help you understand that's how someone somewhere determined what concentration do patients need to receive that is the same solute concentration as the plasma of their blood. And when something is the same, we call it iso. And in this case, tonic or tonicity refers to the concentration of solutes. So this is where I'm going to remind you about tonicity. And you've probably gone through these terms in other biology classes, maybe middle school or high school. Isotonic. Iso meaning the same. What is the same when we say two solutions are isotonic? What is it that's the same? Solute concentration, the number of solutes in the solution. That's what's the same. And so we're going to say that we have two solutions. One solution is the concentration of solutes inside of a red blood cell. The other solution is what's outside of a red blood cell. So if you have a red blood cell and the solution you put that red blood cell in has the same concentration of solutes inside versus outside of the cell, is water going to be transported across that membrane? Be very careful, that's a trick question. When I tell you that, everyone freezes. Water is going to move in and out of that red blood cell. That's osmosis. It's always happening. But when you have an isotonic situation, the net movement of water is zero. You're going to have the same amount going out that comes in. So is that red blood cell going to change shape? No. That red blood cell is happy. Your plasma is 300 milliosmol. The inside of the cell is 300 milliosmoles, so the cell stays the same shape. Now, if we were to give a patient a hypertonic solution, what does the prefix hyper mean? More, above. If you don't know how to remember that definition, babysit a hyperactive kid for a couple hours. You will never, ever, ever forget. More. So hyper, what's more? solutes. Are you getting the message here? Iso, same, solutes. Hyper, more. What? Solutes. So if you put a red blood cell into a hypertonic solution, a solution that has more solutes than what's in the red blood cell, and our example would be putting a red blood cell into salt water, which direction is the water going to move? Into the cell or out of the cell? It's going to move out. Remember, water is drawn to the area of higher solute concentration because water is trying to dilute both solutions equally. So a hypertonic solution has a greater osmotic pressure to draw water out of that red blood cell. And the red blood cell puckers up, shrivels up. Big fancy scientific term is crenate. So should you ever give a patient a hypertonic solution of salt water? No, that would be a bad day, probably your last day on the job. Now we can do the opposite of that, right? What would be the opposite of a hypertonic solution? Hypotonic. What does the prefix hypo mean? Less than. I like to think of hypo as below. Because when you give someone an injection or when you start an IV, what kind of needle are you using? Hypodermic needle. Have you ever heard that term? What does that mean? It's a needle that goes below the dermis of the skin. Hypo meaning below. So now let's put our red blood cell in a solution that has fewer solutes. Let's put it in pure water. No solutes at all. So now we have less solute outside the red blood cell than we do in. Where's the water going to move? Always toward the solutes that are in higher concentration. In this case, water's going to pour into the red blood cell, and what's going to happen? You ever done that experiment with a water balloon where you just fill it with more and more and more, and what eventually happens? It's going to burst. So in a hypotonic solution, our red blood cells are going to fill until they burst. Big fancy word for that is lyse. So do you see why understanding concentrations is important? Again, it's not just something you do in a lab. Every time you put an IV in a solution into a patient, you have to understand the concentration of what's going in because it could have severe repercussions for the cells of the patient, especially the red blood cells. So that's why we have spent so much time talking about water and solutes. Now, as we begin to make strides and steps toward understanding how cells function through molecules and macromolecules, we're going to look at some distinctions here. So we've had our polar molecules, our nonpolar, our amphipathic. Another way we can describe molecules as either being organic or inorganic. Looking at the pictures, looking at the illustrations, what separates an organic molecule from an inorganic molecule? Carbon. Carbon is that atom that's going to help tie other atoms together into what we call organic molecules. And for us, and all life on the planet, and if you've ever watched Star Trek for any length of time or some science fiction movie, we are all carbon-based life forms. You've ever heard that before? That's because we're organic in nature. Now, we can't get away from inorganic substances, right? Because even though we're carbon-based life forms, we are mostly inorganic molecules. And which inorganic molecule? Water. We're mostly water, remember? And that's inorganic. So it's kind of crazy we call ourselves carbon-based life forms, but in fact, it's the carbon, the organic molecules that cause us to function and utilize the water within our bodies. So before we start categorizing and understanding our four major types of macromolecules, I want you to understand how at least three of them are assembled. They're assembled much like when you're playing with Lego blocks, and you take the individual blocks and you put them together into structures, pictures, objects. These macromolecules are principally assembled using monomers, single molecules, small molecules, building blocks. And many of these are going to be linked together into longer and longer chains that we call polymers. And it's a process of polymerization. You probably heard that term before. And in our biological systems, we can build up polymers by the addition of these monomeric units, but we can also do the opposite. We can take a polymer and begin to break off monomers as we need to use them. This is part of our metabolism that we're going to get into later in the semester. You might think of this like stringing pearls on a necklace. That's how these molecules are assembled, one component at a time. So I said we can build up or we can break down. And not surprising, many of these chemical reactions that either build up monomers into polymers or separate monomers from polymers utilize water. Again, another reason for understanding water. And so when we need to link monomers together to make polymers, we are going to take proteins called enzymes, and we're going to get to those here in just a few minutes. But these enzymes are going to assist in forming a chemical reaction that will take ends of these monomers and put them together. And in doing so, they're going to remove two hydrogens and an oxygen. And the result will be a linkage formed between the monomers, and you will liberate a molecule of water. Now, either this morning or last night, you probably took a shower. And with it being a little cooler outside, when you step out of a hot shower, what do you see on the mirror? Condensation. There's water vapor in the air, but when that water vapor hits that cold surface, it condenses. You form water. Guess what this reaction can be called? A condensation reaction when you link the monomers together and you liberate a molecule of water. It's also called a dehydration reaction. It's a linkage. And again, many of our macromolecules are going to be assembled this way. Now, if you understand this illustration, then the opposite, breaking off a monomer, all you would really have to do is change the direction of the arrows. And that's pretty much what we've done here. Taken a polymer with the addition of water, utilizing enzymes again. You break the water apart, separate the bonds. Now you can separate the monomeric units from the polymer. And again, using water again, here we've got hydro, which we know means water. What does lysis mean? We saw lysis in our previous slide with our red blood cells. To burst, to break. Lysis is to break. So hydrolysis literally means water breaking. You're breaking the water molecule apart and using that energy to break the bond that's holding the monomers together into those longer units. So do you see the building up and the tearing down? Again, this is just a basic concept right now. We're talking in generalities. But we can apply this directly to, again, at least 3 of our 4 macromolecules that are going to be important in biological systems. So water's important, carbon's important. Why do you think carbon is the basis of our macromolecules? What's the valence of carbon? What's carbon's atomic number? Oh, I almost got you, didn't I? I heard several of you, 6. What can you not forget when we talk about valence? The 2 electrons in that first orbital. Got to keep track of those. But carbon has a valence of 4, which means carbon can bond with 4 other atoms. In this case, we have carbon, we have C4, methane. Carbon can make single bonds, it can make double bonds, it can make triple bonds. It can form linear chains, it can branch from those chains, or it can form rings like we're going to see in a lot of our carbohydrates. But it's the 4 missing electrons that makes carbon so well suited to form these larger and larger biologically significant macromolecules. And all of our molecules we're going to talk about are chock full of carbon. But the way they're arranged could result in either a polar, a nonpolar, or an amphipathic molecule. It just depends on what else is associated with our carbon. So now let's look at our larger molecules. We started with atoms. We put molecules together. We've categorized them into polar, nonpolar, and amphipathic. Now, let's put them into even bigger categories. Larger molecules we call macromolecules, and we're going to discuss proteins, nucleic acids, carbohydrates, and lipids. Everything your body does, everything that's a part of your body, we can put in one of these 4 categories. Proteins are going to be formed based on your genetic information coming out of the nucleus. That genetic code is going to be used to assemble monomers in the cytoplasm of your cells into polymers that we call proteins. Some of these are going to be chains, some of these are going to be globular units. Some of what we term proteins are going to be several proteins that are together and bonded together with covalent bonds. And so proteins are extremely diverse. They're not only diverse in their structure, but in their function. Enzymes. We'll get to enzymes here in just a few minutes. Enzymes are critical for assisting chemical reactions happening in all of your cells. But proteins are also going to be structural. We talked about red blood cells when we were talking about our tonicity of solutions. Describe the shape of a red blood cell. How would you describe the shape of a red blood cell? Now, we're not going to get into circulation here, but there's kind of a method to my madness here. Is it a circle? Kind of a circle? Is it just a flat disc like a coin? Oh, I'm seeing some of this right here. It has these little depressions on both sides. The big fancy term is biconcave disc. Doesn't it sound smart? Sounds impressive. You know what I like to think of, red blood cells? A squished jelly donut. Because that's how you eat a jelly donut, right? Okay, how many people eat the jelly out first? Come on, admit it. You squeeze it, eat the jelly first, and then you eat the donut. In a class last semester, somebody said, no, I squeeze the jelly out and throw the donut away. I'm like, what? Are you insane? A red blood cell maintains its shape because of a protein called actin that's inside of the cytoplasm. It holds that shape. Because what do red blood cells do? Transport oxygen. How does oxygen get into the red blood cell? We saw that last time in our slide. What kind of transport that doesn't require energy? Going on that downhill slope from high concentration to low concentration. Diffusion. Oxygen comes into the red blood cell by diffusion. So the shape of the red blood cell is important so that every molecule that transports oxygen can actually be close enough to the cell membrane to actually pick up the oxygen. If the red blood cell were round, oxygen would not be able to diffuse into the middle of that cell, so it'd be wasted space. So do you see how proteins are important for shape? But yeah, the shape's going to dictate the function of that particular cell. Motility. Cells have to move around. Your immune system cells, they rely on proteins and receptors inside the cell and outside the cell to crawl around in your body. Without proteins, that wouldn't happen. Transport, hormones, receptor defense, storage, all of that requires proteins. And the instructions for making the proteins resides in your DNA, in your nucleus. Now, the code that's in the nucleus dictates the order and the sequence of the monomers that are going to be used to assemble into our protein chain. Again, this is another place where we can think of a string of pearls. So as we string our pearls together, they're going to have to go together in a certain sequence. And if you get them out of sequence, the protein's not going to be the right structure, and so it's not going to have the right function. And so for proteins, the whole protein is the pearl necklace. The individual pearls are the monomers called amino acids. And the amino acids are assembled together in a chemical reaction using enzymes called a condensation reaction or a dehydration reaction. And in fact, the bond that forms between our amino acids is called a peptide bond. It's a special one. And that's why small proteins are often called what? Peptides. You ever heard that? Polypeptide? That's a reflection of the bond that's holding the monomers of proteins, amino acids, holding them together in the right sequence, and that sequence is dictated by the genetic code of your DNA. Now, proteins can have a lot of different structures. You saw on that slide all the different functions that they have. Well, the reason that the proteins can have so many different functions is because there are 20 different amino acids. The core of an amino acid is the carbon. 4 bonds. There's our carbon. And an amino acid are always going to have an amine group, a hydrogen, and a carboxyl group. Every single amino acid has those 3 components that will be identical. But the last bond of that carbon and amino acid is referred to as the R group. I wish they had made it the V group, because the R group stands for the variable part. And each of your 20 amino acids are going to differ in that R group. The simplest R group is going to be glycine. It has a hydrogen. You get up to some really complex amino acids, tryptophan, phenylalanine, they have these very complex carbon rings. And these R groups are going to provide certain characteristics to the amino acid that will then relate to the function of the protein. We have a whole series of amino acids that are just hydrocarbons. How else can we classify hydrocarbons? What's our word that we put with hydrocarbons? Hydrophobic. So now we've got a part of a protein that's hydrophobic. We also have other amino acids that are polar and charged. They're hydro what? Philic. So certain proteins are going to interact well with certain components of a cell, and some you might find within the plasma membrane. And then we have other amino acids that are going to be present to assist in linking between portions of the protein and making these bridges that can happen. So there's a huge diversity, and we're not going to learn all of these amino acids. I'll save that for the bio majors and the pre-meds. But understanding their properties, especially related to hydrophilic and hydrophobic, is critical in understanding how these proteins attain a certain structure, and therefore the structure of the protein really does dictate its function. Now let's say you have a protein that may be around 380 to 400 amino acids long. Could you imagine a change of one amino acid being that important? Can you envision that? With you have 400 amino acids, 399 are okay, one is messed up. That should be okay, right? You ever heard of sickle cell anemia? Guess how many amino acids are changed in the hemoglobin gene of sickle cell anemia? Do you see the consequences? Amino acids and their properties are important. They're not interchangeable. Because when you change that one amino acid of the hemoglobin gene, it changes the shape of the hemoglobin molecule. And the function is lost. And so as we look at our hierarchy of protein structure, we have 4 levels: primary, secondary, tertiary, and quaternary. The primary level is just a sequence of amino acids. But the sequence of amino acids is going to be important in understanding how does the protein begin to fold, which we call our secondary structure, using hydrogen bonds. There's our chemistry again. We saw it with DNA, now we're using it in proteins. For our secondary structure, hydrogen bonds are either going to cause the protein to coil around itself in what's called an alpha helix, or it may in fact cause the proteins to kind of zigzag back and forth, and another part of the protein come beside it back and forth and make sort of a flat sheet. And that flat sheet's going to look sort of ruffled or pleated. And so it's often referred to as a beta sheet or a beta pleated sheet. Again, amino acids, their sequence and the interaction between amino acids of hydrogen bonds dictates that either coiled shape or the flat shape to provide a secondary level of structure. Well, we got to go even more. When we get to the 3-dimensional look, we're going to see portions that have alpha helices, portions that have the beta sheets, and how the entire protein folds together into a 3-dimensional globular structure. That is our tertiary level. This is where we're going to have other amino acids assisting in forming the folding and our final 3-dimensional structure. Quaternary is a little different because quaternary is where you have more than one protein interacting together, forming covalent bonds into the final functional molecule. We mentioned sickle cell, right? We mentioned hemoglobin. This is a hemoglobin molecule. It consists of 4 protein chains called globin chains. In your body, you have 2 alpha chains and 2 beta chains. If not for every level of organization done correctly, your hemoglobin would not work. You would not be here. Because that is the oxygen transport of your blood. So one word that you're going to hear a lot as we talk about proteins and as we move into enzymes, because most enzymes are proteins, is the term conformation. We say conformation, we're talking about shape or structure of a particular protein. Now, we talked about the beta sheets and the alpha helices forming by hydrogen bonds. Well, you can have a lot of different kinds of bonds. We can have some covalents. We can have ionic, hydrophobic. Some amino acids have a sulfur group. What's the valence of sulfur? How did you remember that? 6 So. See, now we're moving away from just oxygen and carbon. We can do them all based on our little mnemonic. Remember those. But all of those are going to be used to form our tertiary and quaternary structure of our proteins. Now, if our proteins rely on hydrogen bonds, I think you can envision down the road, temperature is going to be pretty important for those molecules, because if they get too hot, what's going to happen to those hydrogen bonds? You'll break them apart. When you break hydrogen bonds and you unfold proteins, we call that denaturing. So this gives you, as far as insulin goes, you'd say insulin is pretty important, right? If you're a nurse dealing with a diabetic patient, you bet you're going to be checking that sugar level and you're going to be understanding how much insulin that patient must receive. Well, insulin consists of 2 protein chains. Here's the sequence of amino acids. What level of organization is the sequence of amino acids? Primary structure. How we fold this individual chain is going to be our secondary, then tertiary structure. Now we have two protein chains tied together. What level of structure is that? Quaternary structure. So when you see two chains tied together, even in a diagram like this, you know we're talking about the fourth and highest level of protein structure. So here's a closer look. The amino acids, and these are sort of the stick diagrams. You can see the dotted lines, and anytime you see dotted lines relative to atoms, what bond are you thinking about? Why do they make them dotted? They're the weakest of our bonds. They're the weakest of our bonds. Now, for DNA to be separated, that's a reason we have those weak bonds. But if proteins have to maintain their folded structure, why do you think we have the weakest of the bonds? Why don't we have the stronger bonds holding these things together? You wouldn't be able to fold it in the first place. You've got to be able to fold it in the first place. Some proteins are also going to have to be flexible. They have to be bendable. And you're not going to have any bend whatsoever if you've got that covalent concrete bond in place. So hydrogen bonds provide a little bit of flexibility to our proteins. Here again is an example of our hemoglobin. 4 chains together. The little lifesaver structure that we see in the middle, that's a group of amino acids called the heme group. And when you talk about blood and circulatory system, you're going to understand that those amino acids of the heme group bind a molecule of iron, and that molecule of iron is in fact what transports the oxygen. But if you don't have the amino acids in the right place because your structure is not folded correctly, are you going to bind the iron? No. If you can't bind the iron, are you going to transport oxygen? No. Do you see the importance of protein structure as it relates to your patient? Now we mentioned temperature. Have any of you ever done the experiment where you take a normal functional protein that has a certain physical property and you denature it and you see it convert its physical properties in a very visible way? Have you ever done that? Everybody shake your head, yeah. If you have cooked an egg, you have done this experiment where you take a protein that's normally folded in a certain way, you denature that protein with heat, you unfold it, and that protein looks very different. When you crack an egg in a skillet, what does that egg look like? When you crack an egg in a skillet, what does it look like? There are 2 parts of it. You got the yolk, which is the yellow, which is the most tasty part, but we're not supposed to have that. What's the other part look like? As soon as you crack it, what's it look like? It's clear. But we call that part the egg what? White. It's clear. Why do we call it the egg clear? Why do we call it the egg white? When you cook it, it's white. That is almost pure albumin. The protein albumin makes up the egg white. And when that protein is folded appropriately, there's enough space between those protein strands for light to pass through. It looks clear. But when you heat it and it turns white, you have unfolded all of the albumin proteins. They all get tangled up together. They take up more space and light can no longer pass through. So now instead of being clear, it's opaque. You would probably agree the physical property before you cook the egg white and after is very different. In addition to being it clear or white, what other physical properties change? It's what? Yeah, the initial white is very runny, but when you cook it, it's a solid. So again, do you see how the shape of proteins can affect physical properties? Every protein in your body has to be folded a certain way or you will not have function and you'll get a very different result at the end. That's why gene mutations can be so devastating. One nucleotide DNA change can result in one amino acid change, which can be total loss of function. All right, so now proteins. Let's move to nucleic acids. Nucleic acids live in your nucleus. This is your genetic code. This is going to be the instruction manual for everything you are, everything you think, everything you do. And when we look at our nucleotides and our nucleic acids, we're going to see that they're composed of monomers that are assembled into polymers. You see how we have a common theme? Building blocks assembled into larger units. What were our monomers of proteins that we just talked about? There are 20 different ones of them. Amino acids are assembled. They're the monomers that are assembled into the polymers we call proteins. For nucleic acids, our monomers are called nucleotides. And here we can see an illustration of a nucleotide. It consists of a phosphated sugar. And for DNA, what is the sugar that is a part of our DNA molecule? Deoxyribose sugar. It's hard to believe that your genetic code is sugar. I like to think of sugar in a very different way, like in my jelly donut. But your DNA is sugar. And the sugars are linked together by phosphate groups. But the important part that we think about are the bases. And we see the base, the nitrogenous base, a lot of nitrogen is present here. And so it's the nitrogenous bases that we pay attention to when we talk about nucleic acids, because it's going to be those bases that hold your two DNA strands together, but it's those bases that make up the code. Morse code. What's Morse code composed of? Dots and dashes. But the number of the dots and dashes and the order of the dots and dashes can make up letters and spell out words. So Morse code has an alphabet of dots and dashes. Nucleic acids have alphabets of 4 nitrogenous bases. And these nitrogenous bases are grouped into what's called pyrimidines and purines. Now you see we have 5 listed. I said 4, but we have 5. Uracil is a nitrogenous base that is found in RNA, not in DNA. But it's still going to be considered a pyrimidine. Now, in addition to information, you know I like to teach you ways to remember the stuff, right? So when you look at our different bases, you have 3 of them that are one ring and 2 that are double rings. So these are small compared to the others. Crazy way to remember this, but when I look at small versus large structure, I look at short versus long name. Now remember they're the opposite. So the purines, the shorter word, are the bigger structure. They have the double rings. The pyrimidines, the longer name, they're the single rings. They're the smaller molecules. Okay, well that helps with the structure. That doesn't help me with the look, right? Well, when I think of pyramids, pyrimidines, I think of all those big blocks of limestone and granite. Those people had to build and quarry and haul and assemble. I think of all those blocks having to be cut. C-U-T. Cytosine, uracil, thymine. So pyramids have to have the blocks cut. That gets your C-U-T. How do you remember the other 2? That's what's left over, right? A and G. So if you remember pyramids cut, automatically you know the A and G goes with the purines. And because of the length of the word, you know the structure's the opposite. Might take a little bit to get that down, but it's pretty simple. And the structure of these bases is important because when you assemble them and hold the chains together, you have to have a pairing of a purine and a pyrimidine. You can't have two purines because that would be too big. There's not enough space. You can't have two pyrimidines because that's too small. They wouldn't reach across. So you must have a pairing of one purine and one pyrimidine spanning that distance. And our pairing is always going to be AT, CG, until we get to RNA. And in RNA, that's where we see the uracil taking the place of the thymine. You only see uracil in RNA, and you never see thymine in RNA. And I also included the equal sign and the triple. What are these representing? What kind of bonds that hold the strands together? Hydrogen bonds. The AT pair has 2, the CG pair has 3. So which pairing is going to be harder to break? The CG, the 3. Takes more temperature to break a CG pair than it does to break an AT pair. Oh, my favorite, carbohydrates. What would life be without our carbohydrates? Well, we wouldn't do much because carbohydrates is where we get most of our fuel. And we get most of our enjoyment from carbohydrates. So carbohydrates, guess how they're assembled? Monomers into polymers, just like we saw with amino acids into proteins, nucleotides into nucleic acids. Now our carbohydrates are going to be assembled, and their monomer is called a monosaccharide. That's nice that they did that. Monomer, mono. Saccharide means sugar. And the way we represent typically a chemical formula of a carbohydrate is with a very simple formula, CH2O, and then we have a subscript N. N simply represents how many carbon atoms are a part of that carbohydrate. And I'll go ahead and introduce this now. We've got some slides coming up, but glucose has 6 carbon molecules. And using this generic formula, what would be the formula of glucose with an N of 6? C6H12O6. See how that works? So if you remember that very simple formula and no carbon, glucose has 6 carbons, I mean, that'll take you far. Now, if we take 2 of our monosaccharides, we use enzymes, we link them together and liberate a molecule of water. Again, there is our condensation reaction. We put 2 of them together, we form a disaccharide. Sucrose, table sugar, disaccharide. Lactose, we have in milk, disaccharide. Now, if we take more than 2 or even a bunch, then we call it a polysaccharide, just a long chain. Glycogen. We've already talked about glycogen in your muscles, in your liver, the storage form of glucose. That's a polysaccharide. Starch. That's how plants store glucose. That's the polysaccharide in plants. So this is our ring formula for glucose. We also have others, galactose, fructose. It's not important that you know the structure, the names of all these. But here are our nucleic acid sugars. Ribose sugar. Where do you find ribose sugar? Which nucleic acid? RNA, deoxyribose DNA. And then here's our sucrose, lactose in milk, and maltose, a different type of disaccharide that we probably encounter in our diets pretty regularly. So just to bring home the point, glucose, 6 carbons. Next week, when we get into respiration and metabolism, we are going to look at the breakdown of glucose into 6 molecules of carbon dioxide. It's going to be important to understand glucose. Why? Well, I mean, it's the most common monosaccharide. It's where we can directly plug into our cellular respiration metabolistic machinery to produce energy. We've mentioned glycogen, the storage form of glucose for animals, particularly for humans. When we bind it together, and we form glycogen. We call it glycogenesis. When we need more sugar in our bloodstream, what hormone, by the way, are we going to be producing that's going to cause the glycogen to undergo glycolysis, glycogen breaking? See that word? What hormone are we going to use early in the morning when we wake up to cause glycogen to break off glucose molecules and put it back into our bloodstream? Glucagon. See, that's one of those words that sounds a lot like everything else. Glucagon. Those alpha cells in your pancreas make that glucagon. All of this is moving, changing, getting that carbohydrate from one place to the next. Here we see our long branched polysaccharide glycogen. Glycogenolysis, break off of glucose, adding glucose, glycogenesis. Remember, these terms are relative to glycogen. Don't call this glucogenesis or glucolysis. That refers to glucose. Again, everything's starting to sound somewhat alike, but you have to keep the terms separate. Glycogen, animals, starch, and cellulose in plants. Cellulose, what does cellulose do in plants? Have you heard of cellulose and what it does in plants? Cellulose is the cell wall. It's hard for me to believe when I look at the redwood trees in California that what's allowing them to stand as tall as they stand is glucose. And the arrangement of glucose strung together into cellulose. But these long strands and very, very tightly woven together makes for a very strong molecule. But I think you can see the branch structure of starch and the highly branched structure of glycogen makes it much easier to break off a lot of glucose molecules over here for cellulose. You'd have to break off the ends. That's, again, one of the reasons that cellulose in the cell walls is very, very stable. Cellulose is not much good to us, right? Fiber helps with your digestive regularity, but it's not very tasty. But I guess it is important for what we do.